Hello students, welcome to week 11 lecture 3. So it will be done in two parts. So today we will doing the first part. So this lecture is mainly about the phylogenetic, metabolic or metabolomic you can say, and comparative analysis of bacteria prokaryotes. Right. So we have already done and touched upon the phylogenetic analysis in the previous lecture, but here we will look at the same thing in context of a bacteria, and then we will look at how in the part 2 how we can do the metabolomic and comparative analysis, metabolic comparative analysis using a case study also. So phylogenetics, as we discussed earlier, also is the study of evolutionary relatedness among organisms. Okay, so basically we can use the molecular or morphological data for this, and we now know that the molecular data works well because it is reproducible and it is not subject dependent and right, subjective. So we rely on the molecular data for this, and through phylogenetics we can know the relationship between whatever exists around us, all the it can be grouped, like for example birds or animals, or even any animals like dogs may have their own phylogeny, right? So this way you can connect the organisms around us using any of the selected molecular marker. And in the context of bacteria, you can imagine there are like billions of bacterial species, and we have only named only 0.5 or 1% of them. They are mainly unculturable, but whatever we could learn about them using the 16S ribosomal RNA marker or any other suitable marker, so that way we can name a bacterial species using phylogeny and also place it in respect to the other bacterial species which are related to that. So that will have a lot of advantages in terms of first of all, we can call that bacterial species by name, and then whatever it contains we can also correlate and connect and compare with the related species or strains or in the genus, or going up in the taxonomic tree. So it is very important, like taxonomy is one of the aspects that we discussed to study the evolution. Like we can see that how that particular species evolved in context of its related species. It could be blue peacock versus green peacock, or it could be Bacillus subtilis versus Bacillus cereothermophilus or Sirius, and like that, right? To compare their genomic regions, to look at the like how the the conserved as well as the specific genes, species specific genes change in that. Okay, in the ecological perspective, that where they exist and what they need from each other. So basically if we know the tree, then we can know that in a given geographical location these are the variants of this particular genus in terms of species are existing, like that. We can see the coevolution of host pathogen like bacteria versus a host, or it could be a bird versus a virus, and like that. Okay, in a particular geographical region, what are the different biological beings that exist, when they diverged, and what is the divergence time, more applicable for basically the eukaryotes, right? And of course medicine, because that way we can know that which are the nearest organisms, or which can for a particular pathogen, or for in terms of host, like when we are looking for something where we can also perform experiments and see the various features and disease resistance, etc. Okay, so the first thing in a phylogenetic tree is to have a sequence alignment, because that actually provides information about how the species are related. Like here also you can see like some positions are very conserved. Okay, some positions like this is a gap, okay, which exists. Some positions are variable. Okay, so this is very important to know, because those basically this tells about how the sequence evolved, changed, and this can be analyzed in three main different ways in terms of phylogenetic analysis, and we can use that information for the constructing the tree. Okay, so basically the alignment tells three important things: what is inserted or deleted from a position, and what is substituted at that position. Okay, so a tree looks like something like this. For example, this is a tree of life where we have the three kingdoms, okay, bacteria, archaea and eukaryota, and basically here it's a tree that is made for a in a context where it is showing a small clade of fish and like evolution of a particular character such as venom in this case, where you can see that it is not present across a clade but it is existing, okay, in some of the clades. That means that it evolved multiple times due to the environment or whatever the evolutionary reasons behind that. Okay, and like this is a tree of life where you can see that it shows the relationship between the different type of organisms that exist in that particular kingdom or in context of the entire tree of life, right? Now more specifically, like using the 16S ribosomal RNA, which is a 1.5 kb gene and contains hypervariable regions and conserved regions and present across all the bacteria, you can construct a phylogenetic tree, for example something like this, where like a new species was found, Anoxibacillus, right? And we wanted to show that how it relates to the existing Anoxibacillus species. So the 16S sequences of all the Anoxibacillus were downloaded and aligned, right? And then basically a tree was made, like which is shown here. Okay, so these are some of the parameters like bootstrapping, the method that was used, and where the paper is available, right? So this tells that okay, this new species is basically closer to this clade, and this is how the nomenclature also is justified for that species. That means it belongs to genus Anoxibacillus, and species name can be given based on any standard convention. Okay, so this is one example. And now in a tree, as we also discussed earlier, basically the distance between or this length, or okay, you can press a tree like this, that is okay, but you cannot stretch a tree like this, because this distance matters a lot in terms of the distance of their like evolution distance it represents, right? And this tree can also be expressed in terms of a textual expression like B and C are related, and then is a comma, and then A is next to it, and then D and E comes together, and together basically you can see that A, B, C and then D is separate, and the same thing you can see here. Okay, and in a tree the three important parts are basically the node, root, branch. Okay, this is a branch, this is a node, okay, and this is the root from which the tree originate, for which you can appropriately use a outlier or use a midpoint distance method, right? That we have discussed in detail in the previous lecture. And so these are the species that were supposed to be compared. This is an outgroup which actually helped in assigning root to this tree, and these are the leaves which are basically the present day what you can see in between, like what happened in these regions, right? It is all hypothetical, and these are basically ancestral lineages, you can say. Okay, so again, like a different way of showing the same thing: leaf, edge, internal node and root. Okay, edge can be a branch also. Okay, so this is another alignment which also shows that the regions that are conserved, the regions that are different. Some regions are more conserved, some stretches like this are more conserved, right? Whereas some are very variable. This is missing, okay, and this part is missing in these organisms. So this way we can tell that how the things are related, and this alignment data we use for the phylogenetic analysis. So first of all, you have to, whatever bacterial genomes you want to compare, or any particular variants of a protein also you can compare using phylogenetic analysis. So you have to gather that sequence data, align it, okay, using an appropriate method. Cluster W is one of the better methods for this. Even MFFT, MAFT alignment, or T coffee muscle can be used, right? And once you have aligned, you can use 3 different ways to reconstruct the tree. One is like distance based or character based, which we will see later. So distance based and character based, that is maximum parsimony and maximum likelihood, are the 3 methods that we use for constructing the tree. We have covered this in the previous lecture. So in this lecture I will give you an overview and more in terms of connection to the bacteria, right? So like this is also an example of an alignment which can be taken as input for constructing the phylogenetic tree. So this is generated by ClusterX, which is the interface based GUI based application of ClusterW, which you can download and upload the sequences on that. It will give you a GUI. You can see the sequences and do the alignment there. Okay, so now in the character based methods, basically what are the characters? It could be the nucleotides or the amino acid sequences, and using the scoring matrices, and this you have already aligned them, and you have got scores for the pairwise comparison of sequences by which you have actually arranged them in the order they appear in the alignment, and also adjusted the gaps or insertion deletions, okay, in that, right? So the distance based methods uses this information of the pairwise alignment directly and produces the tree. Okay, so basically it directly depends upon the alignment information. Okay, like this is a matrix that is constructed from A and B, B and C how close they are, and D and C how close they are, and like that, right? And so it does not give too much weightage to the regions that are varying in between, like, because it is comparing from end to end a particular score for the entire alignment. So it is not giving importance to each residue or regions which are more conserved or more variable or are important for the function, right? Whereas in the character based methods we are dealing with the each and every character or some selected characters based on the method that you are using, and which gives more importance to the variations that happen within the sequence. Okay, so variants of distance metrics are neighbor joining or UPGMA method, and we discussed that earlier. So now when you are doing this for a bacteria, so basically you will be taking the, like we discussed about the example of 16S ribosomal RNA. So you can take the sequences of 16S ribosomal RNA from multiple species which are related to a species, or you got the best hit when you align that 16S ribosomal RNA in the 16S ribosomal RNA database, and you got a list of species that were close to you, so you can use that for alignment. Okay, or you can do by blast exercise, you can know that okay, this could be this genus or species, right? And then you download the species of that bacteria from the database, right, for that particular sequence such as 16 ribosomal RNA, and then align them using ClusterW, and the scores and the information that you get that is translated into a distance matrix, and that helps you to construct the phylogenetic tree, that what is close to what. So okay, like that. Okay, so this method does not give importance to, like, it could be in a alignment that this region could be very variable, or okay, this region is missing. Okay, it might have done the scoring based on the gaps, right, negative scoring, right, penalty, but still it does not give importance to, like, the characters and the gaps in terms of what function they would be doing, right? So basically this provides a very limited information, and it is but it is very fast in terms of making the tree. So this method has limited applications, and more true for very closely related organisms. Okay, so again, just to say that it constructs an all-to-one matrix, and then construct the tree, and this may not be your method of choice initially, because mostly like the other character based methods are more reliable. However, this distance based matrix or method is very fast, right? So another method that we discussed earlier and today we are also touching, it is maximum parsimony, where you are looking at the characters, it could be DNA, nucleotides, or protein amino acids in the sequences that you have compared. So in parsimony you are giving importance to only some selected features or the characters, right? Okay, so basically it is trying to simplify evolution and assuming that too many changes cannot occur at a given position, and it is saying only the informative or the selected sites for its calculation. Okay, so it does not assume any evolutionary pattern, and basically that way it oversimplifies evolution, and we will see in the next example also. Okay, so again it may produce many trees based on the method it employs. Okay, so parsimony means basically extreme unwillingness to spend money, or you can basically you are very miser, okay, like that. So and same thing, it that is why the word parsimony here, maximum parsimony. It tries to use only the informative sites and not all the sites in the alignment data that it gets for the to construct the tree. Okay, so it basically assumptions are that mutations are very rare events and do not occur always. So only those which are relevant or informative will be considered. Okay, and okay, this I just discussed that that is why it takes only few characters that are more important. Okay, like you can see here that it is, these are the informative and uninformative sites, and for a site to be informative, the logic is that it should be present at least among the 4 nucleotides, at least 2 should be there, and at least 2 times. Okay, so like in this case we can see like only 2 and 4 are informative, whereas in position 1 it is in different species A, B, C, D, it is all A. So it is a position or site which is not varying. Okay, so that means it is not informative for the analysis for during the under the maximum parsimony. In this example, in the site 2, we have 2 different nucleotides at 2 different positions, so it's an informative site. And but in 3 basically it is more or less conserved, but just one variation. Okay, and in the 4 it is basically the again 2 nucleotides at varying at least 2 different positions. Okay, so these 2 and 4 are the informative sites here, and it will use only those for the calculations. Okay, so basically that is why it is simplifies the evolution, does not consider all the sites, and trees may not be reproducible, although it is much faster than maximum likelihood and slower than the distance based, right? And and it has also like other issues like homoplasy, where you could have like parallel evolved sequences, and it may not be able to use them for the analysis, right? So the last one is maximum likelihood method, where basically the importance is given to all the residues, and a log likelihood score is calculated for each and every nucleotide, or in case of DNA, or amino acid in case of protein. Okay, and this process, since it is compute intensive, it is considering the entire sequence, so it is it will take definitely more time and computational resource for you. Okay, but this method may be the better one among all the 3, right? And is likely to produce a more reliable tree. Only drawback is that it will take a lot of time, and again the input for this is the alignment data and a statistical model that you can choose among the given options in the software, right? And you can also run it online, but you can also install on your system and run it locally. Okay, so it will also create several trees like maximum parsimony, but it will The trees that will construct will be more reliable because it has given importance to all the positions that were whether it is informative or uninformative. And then we also follow the bootstrapping procedure to recalculate, right, so that tree and the positions in the tree, and we know that how many times we can reproduce those branches and the species relative arrangements. Okay, so combining the two together, bootstrapping and maximum likelihood, generally this could be the method of choice for generating the bacterial phylogeny tree. Okay, and the available packages such as Phylip or there are mega can be used to construct these phylogenetic trees. Okay, RAXML is another package for the complete phylogenetic and post analysis of the phylogenies. So it is available here, and we have discussed this in the previous lecture also. So the same thing that we discussed there is can also be applied for the constructing the bacterial phylogeny. Okay, so in this lecture we looked at the concepts in phylogenetics and how they can be applied to construct a bacterial phylogeny, and what are the different types of phylogenetic analysis method, the significance of phylogenetic analysis, and the input for the analysis, which is the alignment data. So basically alignment has to be done well, and you can export the file in multiple possible formats which can be taken as input for constructing the phylogenetic tree, right. Cluster W can be a method of choice for alignment. And for the analysis now, to construct the tree, basically you have three options: distance based and character based, which has two options, which is the maximum parsimony and maximum likelihood. So in terms of speed, distance based is the fastest, whereas likelihood is the slowest, and parsimony comes in between the two. And depending upon the data you have, type of sequences, you can use any of these three methods, either online or on a locally installed application. Okay, thank you. This is all for the lecture.